Hello, my name is Sion, and I am an assistant instructor for the Georgia Tech School of Biological Sciences. And this is a review of the second half of the genetics unit, which focuses on DNA and the actual molecular mechanisms underlying genetics. To me, this is the actual fun part of the unit. I'm a molecular biologist, so this is the part of the unit I actually care about. Chromosomes, Mendel, that stuff, not so much, but this part, yes, this is the fun part to me. I love this material. And then just again, a quick review. I've updated these slides. I've taken the learning objectives off my part just to save space. But again, there's a handout linked in the description that has the learning objectives for you to synthesize and annotate that material. Again, active recall is a really big part of learning in this course just because there's so much information to cover. So with that, let's go ahead and get started. So we get to start with the actual discovery of DNA and how it works. And so there are basically three classical experiments that proved what the genetic material was. Because back when Mendel was alive and playing with his plants, we had no idea what the genetic material was. We just knew that there was some unit of heredity. And so the first thing was the Griffith experiment, which proved the principle of transformation. So basically, we had two strains of bacteria, the S strain and the R strain. Now, the smooth strain killed mice, and the R strain did not. So what did Griffith do? Well, he killed off the S strain, so they were not alive, and he mixed them with the R strain that were alive. And somehow, we ended up with the F strain when they were mixed together. So that meant that, hey, there is genetic material that can be inherited. And because this genetic material obviously came from dead cells, we called this the transforming principle. Because this DNA was coming from, we didn't know it was DNA at this point. Because this genetic material was transforming the R cell into the S cell. And so there's some genetic material stored in the S cell that could be picked up by the R cell to turn it into an S cell. So basically, the Griffith experiment proved that there was some way traits could be inherited and that it can be uptaken in the environment. So there's the existence of some genetic material and it can be transformed. So then we had these scientists, Avery, McLeod, and McCarty, and they said, hey, there's just genetic material that's causing transformation. We want to know what it actually is. And they said, okay, well, when we have this solution of the dead S cells, the live R cells, and we get the live S cells, we can either get live S cells or live R cells. So if we mix something in that destroys the genetic material, we will get R cells because whatever the genetic material is will not be there. But if we mix something in and it does not destroy the genetic material, we will get the S cells, which is the natural outcome of the experiment. So that's what they did. They just mixed a bunch of stuff into this solution, right? So the first thing they said is, hey, is the genetic material protein? I'm going to mix it with some enzymes, some proteases that destroy protein. And guess what? We get S cells. So protein is not the genetic material. What about lipids? Well, we mixed it with some lipases, which destroy lipids, and we still get S cells. So lipids are not the genetic material. And we did the same thing over and over and over and over and over until we added DNA aces, which destroy DNA. And when we destroy all the DNA in solution, we get R cells, which means that whatever was allowing the R cells to become S cells, be transformed, was due to DNA. So I'm just going to highlight this right here. This slide says all you need is that Avery, McLeod, and McCarty proved that transformation is due to DNA. So then we have Hershey and Chase who go, hey, we know that there's this genetic material that allows for transformation. We know it's DNA, but we don't know if that's the ultimate source of genetic material because at the time there were two hypotheses. Either traits were passed down through protein or because of Avery, McCarty, and McLeod, hey, maybe it's due to DNA because it's allowing for transformation. So we're going to prove whatever this material is. So what did we do? Well, we radio-labeled proteins and we radio-labeled DNA. And what we did was we put this radioactive DNA inside a bacteriophage, right? And that bacteriophage injected whatever its genetic material was into a bacteria. So the genetic material was either DNA or protein, and we had them both radio-labeled so we could tell what was what. And once that phage left the bacteria, we centrifuged it down, and we said, hey, what's inside is radioactive DNA, which means that the phage injected its genetic material, and that DNA was the genetic material. Because the belief was, hey, genetic material is either protein or DNA, and it turns out that when the phage injected its genetic material, it was DNA. So Hershey and Chase proved definitively DNA is the genetic material. And there's a practice question based off of this. So what would happen if instead of DNA, protein was the genetic material? Where would the radio-labeled protein be found? Would it be found inside or outside the cell? Well, remember, the phage is injecting its genetic material inside the bacteria. So therefore, it must be, if it's the genetic material, it must be inside the bacteria. So we can rule out A and B. And is it either going to be in the cytoplasm, or is it going to be inside the entire cell? Well, it's not. This question, C, this answer says it's not going to enter the nucleus. Well, that basically violates what we know genetic material to have a function as. So we know that the answer is D, because protein is going to carry the genetic material. So the answer is D. Moving on, we have some fundamental rules about the properties of DNA. So DNA consists of three things: a phosphate group, a sugar, and a nitrogenous base. Now the sugar is either going to be ribose or deoxyribose. That's the difference between DNA and RNA. But if you see here, the nitrogenous base, that's going to be A, T, C, or G. And remember that A pairs with T and C pairs with G. A good way to remember that is apples go in trees and cars go in the garage. You know that when I take my exams, even now, I say that when I write my complementary bases. And if we look here, why can't A pair with G? Well, look at this schematic of DNA. Between A and T, we form 2 hydrogen bonds. And between C and G, we form 3 hydrogen bonds. And that's really important to the stability of DNA. And we see here with this practice question where we have the enzyme helicase that unzips the A and T base pairs, preferably over the C and G base pairs. Why does this happen? Well, let's think. What is the only difference between these bases? And I identified that right here. It's in the number of hydrogen bonds between them. And remember, just kind of intuitively, the more bonds we have, the harder it is to break something apart, right? C and G base pairs are more prone to mutation. That doesn't really make sense. There's nothing about methylation, which is a form of protein modification. And A and T are less stable. That doesn't really make sense. If they're pairing, they're obviously stable. So the answer that makes sense here is that, hey, A and T are held together by 2 hydrogen bonds. C and G are held together by 3. 2 is easier to separate than 3. So the correct answer is B. Moving on, there's something called Chargaff's rule that you need to know. And that basically means that there's a 1-to-1 ratio of A and T and C and G. And obviously that would make sense because every one A pairs with one T and one G pairs with one C. And the good thing about that is that allows us to do a calculation based off of knowing the concentration of one base pair. So we're saying, hey, we have 15% guanine in our DNA, so 15% G. What percentage of thymine T do we have? Well, let's go through and let's do the math. So if we have 15% G, we must have 15% C, which means that 30% of this organism is G and C, and that means that the other 70% must be A and T. Okay? And then what is half of 70%? Because there's a 1-to-1 ratio. So that means that, I'll write this in pink, there must be 35% A and 35% T. And if you add up 35, 35, 15, 15, you get 100. And so that means that there's 35% thiamine. Moving on, Watson and Crick. Well, it's called the Watson and Crick model, but really it should be called the Rosalind Franklin model because they just kind of stole her work. But whatever. They proved that DNA is a structure called a duplex. And it has this anti-parallel nature where one end runs from 5' down to 3', and the other runs from 3' down to 5', right? And on the outside are the sugars and the phosphates, and on the inside are our nitrogenous bases. And what is between those nitrogenous bases? That's right, hydrogen bonds. And again, right here we see anti-parallel, and the DNA strands are complementary. So on one side we have a T, on the other side we have an A, and they run in opposite directions. 5', 3', 3', 5'. And that's going to be the structure of a complete strand of DNA. And so now we know the structure of DNA, we can ask ourselves, how is DNA replicated? And I have this idea written out on the slide because it's really important to understand in reasoning through how it replicates. DNA is composed of two complementary strands, right? We see this here. That's in the Watson and Crick model, which means that one strand must be a template to be replicated from. So let's say we have our strand of DNA here, right? One of these strands must be the template for replication, okay? And so based on that, there were three possible models for how DNA could replicate. We have the conservative model, where we just get one new strand and two new strands, and the old and the new don't get mixed. We have the semi-conservative strand, where the old and new, you get one new strand, one old strand. Or we have the dispersive model, where it is literally completely random how much old and how much new you get. I'm going to draw that out right here. Maybe we have a little bit more purple in this one, some pink. All right, so some old, some new. One old, one new. Or all old, all new. Those are the three models: conservative, semi-conservative, or dispersive. And so how was this proven? Well, it was proven through something called the most beautiful experiment in biology, which I think is really fitting. But what happened was we made DNA that was heavy. And so what does that mean? It has 15N nitrogen instead of regular 14N nitrogen. So we had all heavy DNA. And we then added regular light nitrogen. And so what we said was, hey, depending on how the DNA replicates, the heavy DNA is going to end up somewhere else. So if it's conservative, we're going to get all heavy strands and all light strands. Or if it's semi-conservative, we're going to get half heavy, half light. And if it's dispersive, it's just going to be a random mix of heavy and light DNA. And what we can do is we can separate heavy and light DNA, because they have a different weight, with a centrifuge. And so what do we get? Well, on the first generation, we have all heavy, right? All heavy. Then on the second generation, what happened? We got half heavy, half light. That's in the second generation. And in the third generation, what happened? We got we got one strand that was half heavy, half light, and we got two strands that were completely light. And that makes sense because we're using all light nitrogen from now on. We only have the original heavy strands. And that follows the semi-conservative model, right? Because especially at the first generation, that makes sense. You can see that where we have one new strand that's all, we have one strand that's completely new, one strand that's completely old, and we can tell what the old strand is because it is heavy. This proved the semi-conservative model, but what would happen if it was all conservative? Well, if it was all conservative, we would have one band of heavy DNA and one band of light DNA. And what would happen if it was dispersive? Well, it would be random, right? It would be random levels of density between the fragments. And we can see the semi-conservative model where we have this one band of heavy DNA. It's getting smaller as more bands with light DNA get made. But again, the Mendelssohn-Stahl experiment, it proved that we had semi-conservative replication. So now, let's look at a practice question which says, hey, what would it look like if DNA is replicated by dispersive replication instead? And what does dispersive replication mean? That means random. So which one of these answer choices aligns with the idea that we're going to have a random amount of 14 and 15 in DNA? Well, right here, double helices composed of random amounts of 15 and 14 in DNA, which means that C is the... So now we know the model through which DNA is replicated. What is the actual mechanism for what happens? Well, we make something called a replication fork. And the important idea here is that DNA is split, the two strands, right? So we have one strand, we have two, and they are split apart by this enzyme helicase. And then what I also want you to recognize is that these strands are antiparallel. On one strand we have the 3' to 5', and on the other strand we have the 5' to 3'. What else? Well, DNA is replicated by the enzyme polymerase. Now, polymerase cannot bind on its own. It requires an RNA primer. And what makes that primer? Primase. So if you look at this replication fork, we have the helicase unzipping the DNA, and we have primase creating what? An RNA primer. And what's later going to bind to this? I'm going to draw it right here. We have polymerase. Okay? And I want you to recognize an important point here because this is really crucial how DNA replicates. But DNA polymerase, I only drew it on one side. Why? Because it can only bind to the 3' end of a primer. And remember that if, I need a blank side to do this. But if we have a 5' to 3' fragment of DNA, the primer is going to bind to that in the 3' to 5' direction. Why? Because it has to be complementary. Okay? It's a big idea here. And that's where we get this practice question on how does DNA replication differ between the leading strand and the lagging strand. So we have one strand that's 5' to 3' and one strand that's 3' to 5'. Okay, well, let's see. One says DNA polymerase can only add new nucleotides to the 5' end. Well, I literally said that's not true because recall that polymerase works on the 3' hydroxide. So we know that one is just not true. What about 2? DNA polymerase can only add new nucleotides to the 3' end. That is literally what I said, so we know that 2 is true. Primase C3 says primase is not needed to initiate replication of the leading strand. That's not true. Primase is always needed to synthesize RNA primers. And then lastly, helicase cannot work on the lagging strand. Well, if you just look at this image here, we have helicase going from the one strand, which is the 3' to 5' strand, and the helicase going from the 3' to 5' in the other way. So that is not true either. So the only one that's true is 2. Okay, so now let's talk a little bit more about the function of polymerase. Because this is important, polymerase is a protein, which means that its function is dependent on structures in the environment. And I really need you to understand this first bullet point right here, which is that DNA polymerase only adds new nucleotides to the 3' end. Okay? So we have one strand, the template strand, that runs 3' to 5', and that's called the leading strand. So leading is going to go from 3' to 5', which makes sense because, again, we can only add to the 5' end. So we have a lagging strand, and the lagging strand is where the polymerase cannot directly bind. Why? Because it's going from 5' to 3', and we can only add to the 3' end. So what happens? Well, we have our leading strand here, the helicase is unzipping it, and the DNA polymerase is following right behind because DNA polymerase is going to bind at this 3' end, and it's going to make a new strand going from 5' to 3'. Well, what about the other strand? The lagging strand runs from 5' to 3'. Can polymerase bind to 5'? No. So what ends up happening is we have a bunch of little primers coming in, and the primers have a 3' end on them, right? Because if we're going from 3' to 5', obviously what would end up happening is that we get a 3' to 5' primer. And so polymerase is going to bind to the 3' end of that primer, and it's going to synthesize a short fragment. and that is something called an Okazaki fragment. That's really important to recognize. So hey, I want you to recognize we have the leading strand that goes from 3' to 5' and is replicated 5' to 3', and it goes continuously. The lagging strand goes from 5' to 3', and therefore the complementary strand is going to be 3' to 5' and is replicated in fragments, Okazaki fragments. So now we have this diagram of DNA replication here, and we have about 6 or 7 things that we need to label on it. Okay, and I think the first thing that we need to start with is identifying our leading strand and our lagging strand. So this strand up here, right, we have, it's going from, again, there's a little twist on the end which makes it a little bit tricky, but we have this 5' end right here, and it goes from 5' to 3', and this strand down here goes from 3' to 5'. So which one is our leading strand and which one is our lagging strand? So remember, the leading strand is the one that polymerase can bind to, and polymerase binds at the 3' end. So 2 is our leading strand, 5 is our lagging strand. So what else can we identify? What's doing the unzipping of DNA right here? That's the enzyme helicase. Wow, helicase. What is doing the continuous replication on our leading strand, making our complementary strand? That's right, that is the enzyme polymerase. And there's also a second polymerase, that's why I wrote 4 over here, but yeah, that's doing the replication on our lagging strand. What is 6 pointing to, this little thing right here? That is pointing to what is 6 pointing to right here? This little thing that is bound to our lagging strand. That's going to be our RNA primer, which allows for replication of our Okazaki fragments. Now, one important thing to note that I didn't really mention here because it's not on the slide, but there has to be something sealing this new replicated fragment to the DNA. And that's done by an enzyme that works kind of like I say that it glues these fragments to the lagging strand, and it's called ligase. So what is this little blue thing doing right here? That is a ligase, and it's gluing the Okazaki fragment to the lagging strand. And then I didn't put a label here for this arrow because it's kind of ambiguous. But yeah, we have our lagging strand going from 5' to 3', our leading strand going from 3' to 5'. We have our polymerases work on replication. And how is this lagging strand amplified, or how is the complementary strand made? Through Okazaki fragments sealed up by ligase. Polymerase is able to bind because we have RNA primers, and unzipping is being done by helicase. So now, with this practice question, I can almost guarantee you're going to see one on this like the exam, where there's going to be a defect with part of the replication machinery, and you have to identify what has gone wrong and what's going to be affected. And in this case, what's going to happen if polymerase is not functional? Okay? And the best thing I can do is say, hey, go through your answer choices and identify what enzyme is responsible for what. So in this case, choice A says template strands not being able to separate. What enzyme would cause, what would have to go wrong with an enzyme to cause that to happen? The enzyme helicase would have to be non-functional. Synthesis would result in many small segments of DNA rather than a complete molecule. What is responsible for sealing the segments together? That's ligase. Synthesis of a complementary chain would not initiate. What does polymerase do? Synthesize the complementary chain. So C is kind of sounding right to me. And then let's look at D. The DNA strands produced would not be complementary to the template strands. And D is impossible. So the correct answer is C. So now we have this chart here that I made for you, and it has the 4 enzymes that are necessary for replication. Polymerase, ligase, primase, helicase, what they do, and what would happen if they didn't work. And I want you to go ahead and fill in that chart and identify maybe what you didn't know until you saw this chart. So take a second to do that. And now we can talk about the central dogma of molecular biology. So the idea of the central dogma is we have DNA, the actual genetic material, DNA is transcribed into RNA, which is single-stranded. And this single-stranded RNA is translated to make proteins. What we just learned was of the actual replication of DNA, so now we're going to learn how proteins get made from DNA. So now we're going to talk about the process of turning DNA into RNA, so the process of transcription. And there's actually a pretty big difference between eukaryotic and prokaryotic transcription. So what I want you to do is I want you to picture a prokaryotic cell in your mind. And if you think about it, prokaryotes don't really have much going for them. They don't really have organelles. Everything is just floating around on the inside. So that means that as DNA is being transcribed in prokaryotes, right, that DNA is becoming mRNA, the ribosomes are going to go ahead and bind to the DNA and begin to translate it. So in prokaryotes, that process of transcription and translation is occurring simultaneously. Okay? We have simultaneous transcription and translation because everything's floating around. As soon as the first RNA, the mRNA, gets made, a ribosome can bind and begin translating it. So in eukaryotes, there has to be this additional step of RNA processing during transcription. And why that is because if you look at a part of eukaryotic RNA, we have the coding sequences called exons, and we have non-coding sequences called introns. So introns don't really do anything. They just sit there and take up space. And there is no point in turning those into protein because they don't code for anything. So what has to happen? The introns have to be spliced out. So eukaryotic transcription goes through three main steps of processing. We have the splicing step where we remove the non-coding introns. Then we slap on this 5' cap, which is necessary for translation to start. And then we add on this long chain of A's called a poly-A tail. So those are the three main steps, right? And I want you to pay close attention to this bullet here. During the splicing step, we can splice the introns out in a different way so that we get a different arrangement of exons. That's called alternative splicing. And that's very important because it allows for genetic diversity in eukaryotes because the exons can be arranged in different ways. So again, recall those three steps with me: splicing out of the introns, attachment of a 5' cap, and then slapping on this poly-A tail at the end. So then, how does translation take place? Well, we need three types of RNA in order to turn the DNA into a protein. First, we have messenger RNA, mRNA. That is what actually carries and codes for the protein. So mRNA is made from our genomic DNA, and it's used to go to the ribosome and code for proteins. Well, what actually happens in the ribosome? Well, the ribosome itself is made up of rRNA. And the ribosomes bind to the mRNA, and they make the protein chain, right? So we have our mRNA, which codes for the proteins. It comes to the ribosome, which is made up of rRNA. And we get this chain of proteins being built. But what is between the mRNA and the chain of proteins? tRNA, which stands for transfer RNA, because it brings the amino acids to the ribosome and actually allows this chain to be built. So we have mRNA coding for protein, rRNA coding for ribosomes, tRNA coding for transfer RNA that brings the amino acids to make a protein. So what starts and stops the process of translation? It's not random. Something has to tell the protein chain to start being made. That's called the start codon, okay? And the start codon is going to take place at the 5' end of DNA, and that's going to tell the polypeptide chain of amino acids to start being created. The start codon is universal. It's methionine, and it's coded by AUG. So on your exam, you're going to get a question, and it's going to be like, what is the protein sequence made by this DNA? Well, when you look at the mRNA, don't translate anything before AUG. Don't translate anything before your start codon. It's always going to be AUG that starts your protein chain. It's always going to be MET. So if it's like, in your answer choices, just look for MET, blah, 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 because it always has to start with your start codon. Now, There's also a stop codon, which tells the chain to stop, and it's coded for by multiple codons. So you'll have a codon wheel or a chart or something that'll tell you what your different stop codons are, because there's not just one. And then how does translation actually happen? Well, you have some word vomit here that tells you how it happens, but we start AUG, methionine, at the 5' end of the mRNA. Then we move down in sequences of 3, our codons, in order to bring amino acids to form a polypeptide chain. And what does that bringing of the amino acids? tRNA. And here's a graphic of that happening, right? We have our ribosome made up of what? Our RNA. It's translating the mRNA by bringing amino acids to each codon. And what's doing that? tRNA. And there's a little problem here to me. Why are we starting with UUU coding for lysine? What should we be starting with? Methionine, coded by what? AUG. So this graphic is a little bit wrong to me, right? Because we should be starting with a start codon. There should not be a protein chain that starts without methionine. So here's a practice question. And guess what? This is just like something you would see on the exam. So we have this mRNA strand, and we're asked what would the first 3 amino acids be? And we're given a wheel of what codons correspond to what amino acids. So let's look at this strand. And what did I say you need to do? You need to identify the start codon, right? Because that's the only way for the protein chain to start getting built. And what do you know? We know that the start codon is methionine. So we can rule out the 2 answers that don't have methionine. And then we can go ahead and look at this mRNA strand. Where is the start codon? AUG, coding for methionine, right here. So we have AUG. Whoops. We have AUG. Then we have GCC, ACA. What does GCC code for? Well, let's see. We have a G, then we have a C, and then we have another C. That codes for ALA. So we have MET, ALA, and what does ACA code for? First letter is an A, then we have a C, and then we have another A. That codes for THR. So the first 3 amino acids are MET, ALA, THR. So don't get mixed up. Your first amino acid has to be methionine. Okay, I want you to recognize that. That's a really big takeaway. And here's a little note from me about that place a little north of here, which is when stop codons were first discovered, they were called nonsense codons. Think nonsense mutations. And if you look here, one of those stop codons just happens to be UGA. So scientifically, UGA is nonsense, and they can stop barking at us. For the love of God. Okay, so there are also 4 different types of mutations, okay? So the first type is called a synonymous. I will never be saying that. I'm going to say a silent mutation, and it has no effect. So a silent mutation is just like the swapping of maybe, let's see here. Let's go back to the codon chart, right? If you see here, there are 4 different ways to code for ALA. So maybe this A here gets swapped for a C. That's a silent mutation because we still get ALA. There is no change. So there's no change due to a silent mutation, hence the word silent. Now a non-synonymous is also called a missense mutation, and that is a single nucleotide change, right? So maybe instead of getting GCG coding for ALA, we get GAG. So the C got changed to an A, and we get glue. That is bad. That is a change. So a single nucleotide change is a non-synonymous mutation. Okay? Now, we have two really big mutations. So silent and non-synonymous don't necessarily have to be completely destroy the protein we're making. That is not true. However, a nonsense or a UGA mutation is, because if we get something that codes for a stop codon earlier than when it does, that protein is just going to stop right there. And if you have a protein that's not done being built, it's not going to be functional. So a nonsense mutation is very bad. But what might even be worse than a nonsense mutation, even though nothing can really be worse than UGA, is a frameshift mutation, right? So a frameshift mutation is going to shift how the codons are read. So for example here, we had the insertion of a T, and what happens? Instead of reading ATG, we read ATT, and that G is pushed to the next codon. So we could get an entirely different protein. And what that's going to be caused by is either insertion or deletion of a nucleotide that shifts how the codons are read. And that's going to alter every amino acid downstream of it. So a frame shift and nonsense mutation are horrible because you're going to get, likely, extremely likely, you're going to get a I keep using the highlighter. You're going to get a non-functional protein. So these two, nonsense, frame shift, extremely bad. Silent, there's going to be no change, so it's not the end of the world. Now, non-synonymous mutation. That has a degree of how bad it can be in terms of the protein. So for example, if you get one polar amino acid changed to another polar amino acid, that's not that bad because they can kind of take place of each other. But if that polar amino acid got swapped with a non-polar amino acid, that would be very bad because you had a change that fundamentally changed one of the building blocks, one of the amino acids of the protein. So it's just important to understand the 4 different types of mutations here. And now we get to talk about how genes are regulated. Okay, so what's pretty important to understand is that obviously we have genes coding for proteins. But of almost equal importance is how much protein gets made, which is why we have ways of controlling or increasing or decreasing the amount of protein that gets made. So key thing to understand is that a promoter is a site where polymerase is going to bind. And so we can either increase or decrease the ability of polymerase to bind to a promoter, right? Which is what are called transcription factors. So we have positive transcription factors, which increase transcription. We have negative transcription factors that decrease transcription. We see that right here. We have this enhancer that increases the ability of polymerase to bind to this promoter. We have this silencer or this repressor that alters the ability of polymerase to bind to that promoter. And we also have something called a constitutive transcription factor, which is always expressed, always on, always. The takeaway here is that all organisms are going to have some way of some transcription factors that alter that promoter to either increase or decrease gene expression, or they're co-regulated, so regulated by multiple genes. Just again, recognize, hey, we can increase it, we can decrease it, and all organisms have some form of being able to do that. So we see here, in talking about the types of promoters, what's more likely to be controlled by a constitutive promoter rather than a regular promoter. And you see it in bold, but constitutive, right? It means always on. So what always needs to be on, right? Because organisms do not want to waste time making proteins they don't need. For example, a gene encoding antibiotic resistance protein. Well, when do you need antibiotic resistance protein? In the presence of an antibiotic. So do you always need to be making that? No way. That is a waste of energy making that protein. What about enzyme encoding lactose metabolism? Are we always needing to metabolize lactose? Probably not. What about protein for heat shock that is needed during high temperature stress? Well, the fact that it says stress should be clue enough that we don't need that. But what about polymerase, which we need for transcription in all conditions? All conditions. We always need that. There's no reason to not be making that. We need a constitutive promoter for that. So in prokaryotes, we have this process of co-regulation. And co-regulation is achieved through something called an operon. So we have multiple genes put next to each other. 1, 2, 3, right? And then we have our promoter, like we said, which is where polymerase binds. So multiple genes under the control of a single promoter. And what that means is that when RNA polymerase binds, all these genes are going to be translated into proteins. So these genes are co-regulated together because they're under a single promoter. So operons have three different parts, right? So we have the promoter, which is where RNA polymerase binds, right? Then we have our genes, right, that are actually co-regulated. But we have two things. We have an operator, which is where a repressor can bind, right? You see that right here. Operator, where a repressor can bind, and an activator, which promotes the binding of RNA polymerase. So in this case, it's called a cap site, but this is an activator that promotes RNA polymerase binding to this operator, whereas this repressor, it would physically stop RNA polymerase from moving on. So the operator basically is going to control whether or not this gene can be transcribed because it's blocking polymerase, The activator is just to say, hey, we need more binding of RNA polymerase. So just get those definitions locked into your mind right there. Because now we can go ahead and look at this model system called the lac operon. So the lac operon is responsible for the metabolism of lactose. E. coli don't really want to metabolize lactose, they want to metabolize glucose. But because sometimes they're not always going to have glucose, they need a way of metabolizing this other sugar, lactose. So there are only two situations the E. coli can find themselves in. No lactose with lactose, right? So what happens when there's no lactose? Do we need to be coding for these genes responsible for metabolizing lactose? Absolutely not. So when there is no lactose, guess what? The operator, the repressor, is going to bind to the operator and prevent transcription by RNA polymerase. So no lactose, no metabolism. What about with lactose? With lactose, something is going to bind to this repressor, bringing it away, allowing RNA polymerase to move through and transcribe these lactose metabolizing genes. You may also see a model system called the trip operon, which is responsible for metabolizing or creating the protein tryptophan. But yeah, these are just model systems that are important for how operons work. So remember, we have the operator where a repressor binds. Repressor becomes unbound due to the presence of lactose, allowing RNA to transcribe the lactose genes. So now we're given a practice question about this where we're told we have both glucose and lactose, and we're asked for what's going to result in the strongest activation of the lac operon. Okay, so really, what are we saying here? Well, we're looking for what is going to promote the activation of the activator, right? Think back to the structure of an operon. So we need to promote the activator. And what's going to do that? Well, in the lac operon, the activator is called CAP. So we need CAP to be active. So we can go ahead and rule out A and B because what? They say CAP is inactive. And CAP is our activator. So what does what does C and D say? CAP is active, right? Then what happens that in the second part? Well, we have the repressor, right? And we need high lactose to make the repressor inactive. Because when there's low lactose, the repressor is going to be active. But high lactose pulls that repressor off. So if we have an active activator, the CAP site, and we have an inactive repressor, all those lactose genes are going to be transcribed and expressed. So the correct answer here is C. And now, again, here's a chance to label an operon just to cement that knowledge. So what I'm going to do is I'm going to lump 6, 7, and 8 together because those are just our co-regulated genes. What about 2? What is 2? And what is 2 binding to? 2 is binding to our operator, which must make 2 a repressor. And if you look at the second image here, what is 1 binding to? And what is 1 doing? Well, 1 is transcribing these genes, so 1 is RNA polymerase. And what does polymerase bind to? A promoter. Okay? And you can see the difference here. 5 is lactose, and it's pulling that repressor away, allowing RNA polymerase to metabolize or to transcribe these genes, right? So, yeah, that is the lac operon labeled. So eukaryotes, well, we have a lot of genes in a lot of different places, right? We have all these different chromosomes. And so in eukaryotes, when we co-regulate things, there's one big similarity we all have. And that is co-regulated genes have the same regulated element, even if they're super far away. So 2 co-regulated genes can be on completely separated chromosomes, super far away, but they have the same regulatory element, and that makes them co-regulated. Which you have a kind of intuitive sense for, right? Because if it's the same regulatory element, the control of that is going to be the same. So then we see this structure here of chromatin, right? And chromatin is DNA. I keep doing that with the highlighter. DNA packed around protein, right? That's what chromatin is. And when we have that, when we have tight packing, can we transcribe that DNA if it's super closely packed together and wound tightly? No, it is inaccessible. I'm going to highlight that actually. Tight packing equals inaccessible DNA. Loose packing equals accessible DNA. Accessible DNA can obviously be transcribed, right? So when we take that DNA, right here, it's very tightly packed, and we loosen it up, then we can transcribe it, and that's how we get protein from it. So basically, that's what this bullet is saying here. Tight and loose packing of DNA is one, caused by protein modifications, and two, regulates expression of genes. Because when DNA is tightly packed, can we express those genes? No, we cannot even transcribe that DNA. But when it's loosely packed, yes, we can bind to it, we can transcribe it, get mRNA, make those genes. And that's an important distinction to recognize. Now, here's something I also want you to recognize. If we look at the size of eukaryotic genomes, we go from 10 to the 1 megabases, 10 to the 4 megabases. That is over 10,000 times different of a eukaryotic genome size. Compared to bacteria or archaea, what is going on? Why is our genome size so large? And if you think about it, what is the difference between a prokaryotic and eukaryotic genome, right? Well, in prokaryotic genes, I've just written this table out for you, right? Prokaryotic genomes, all genes have a function. Everything codes for a protein, and it's going to be in a singular circular chromosome. A single circular chromosome of all functional genes. Therefore, the size of the genome is directly correlated to how metabolically complex a prokaryote is, because every gene has a function. What about eukaryotes? Eukaryotes have introns, so non-coding sequences. If you get to hear my 5.1 recombinant DNA lecture, I go on a tangent about introns. But anyways, we have these non-coding sequences, which means that size in eukaryotes, size of the genome, does not equal complexity, because you can have 1 million introns that don't do anything for you, which gives you a super large genome, but it doesn't make you have any complex functions. Now, eukaryotic genomes also have chromosomes, and each chromosome has this duplex DNA molecule, right, packed around a histone, right? That's that chromatin structure. Eukaryotic genes also don't have operons, and mRNA makes one protein, right? We don't have this multiple genes situation like we see here in prokaryotes. So again, talking about eukaryotic genomes, a fun fact I like to think of is like humans have around 40,000 genes, and rice has about 55,000. So rice has more genes than us, and in a way, like, you hear that and you're like, that makes no sense, because humans are way more complex than a grain of rice, right? And we actually see that because humans have introns, right? Introns make up 25% of our genome. And introns don't code for anything, but what do they allow? Alternative mRNA splicing. That is very important. We can have fewer genes than rice, but we can use alternative splicing, putting those exons in a different order to produce multiple proteins from a single gene. Rice don't really do that as much. So because of the presence of introns taking up so much of our genome, we can, when we splice this out, we can put our exons in a different order and allow for that process of mRNA splicing. And you can see here the exons that directly code for proteins are less than 2% of our genome. And then we also have something called transposons, which are these genetic elements that can move around, but we don't really know, or we have no real function for them besides like ancestry. And that leads us to the point that I made on the last slide, which is that genome size does not equal metabolic complexity in eukaryotes. And then we have this practice question about rice, where rice has a genome of 430 million base pairs. The human genome is 3.2 billion base pairs. What a crazy difference. But rice has a higher proportion of genes. Why is this? So we can look at the answer choices and see what makes the most sense. And if you can recall the basic fact, hey, humans have a crazy amount of introns, all these non-coding sequences, which reduces the amount of protein coding DNA we have, right? It's only 2% exons, which means that B is the correct answer, right? And the rest make a little sense when you read them at first, but B is the best answer. And so that is a review of lectures 5 through 8, which covers DNA and the actual molecular mechanisms underlying expression of these traits. And so until next time, best of luck with your studies.